within 30 seconds. Within 30 seconds only.
Okay, sir. Nagarkar madam, are you ready?
Yes, ma'am.
Okay.
So, okay now. And I request everyone to mute your mic. I request everyone don't disturb, and I request Retno ma'am to type your email ID in the chat box.
Already, Dr. Desmond.
Very good. Thank you. Thank you. Thank you. So now, Sarita, you can proceed.
Yes, sir. Shall we start now?
Yes.
Thank you, sir. Am I audible?
Yes, yes, you are audible.
Okay. So greetings to one and all, and a very good morning to all the excellencies. It gives me an immense warmth and great pleasure to welcome you all for today's international webinar on the potency of Streptomyces thermocarboxydus to combat Fusarium oxysporum, the cause of leaf rot disease, on allobarbendensis mill in Bali, organized by Department of Microbiology, Sanjeevani Arts, Commerce and Science College, Kopargaon, and Microbiology Society, India. Be the change you want to see in the world. By this quote given by Mahatma Gandhi, I, Nagarkar Rachna, would like to begin our today's session. So, on behalf of whole organizing committee of Sanjeevani Arts, Commerce and Science College and Microbiology Society, India, it gives me tremendous contentment to welcome the most esteemed personalities. So, first of all, I would like to welcome our today's winner speaker, respected Professor Dr. Retno Kauri from Biology Department, Faculty Mathematics and Natural Sciences, Udayana, University, Bali, Indonesia. It is an honorable moment for our college to have your prestigious presence and valuable talk. Then I would like to welcome respected Dr. A.M. Deshmukh sir, President of Microbiology Society, India. We are very grateful and delightful to have you for this session and also for providing us such a great platform for such an interactive session. I would also like to welcome Dr. Pattinathu. There are some faculties from other colleges who have joined, so I welcome all. Then I also welcome Dr. Buttada ma'am, Head Department of Microbiology, all staff members and all the attendees present over here. So once again, I welcome you all on behalf of Sanjeevani College. So now I request our of today's webinar. Thank you. Over to you, ma'am.
Thank you, ma'am. Am I audible?
Yes, ma'am.
Okay. Good morning to all. We, Sanjeevani Arts, Commerce and Science College, Kopargaon, in association with the Microbiology Society India, has organized international webinar. The motto of organize this international webinar is to provide the research exposure to the students. Nowadays, due to the increasing competition, it is very essential to update ourselves, and the students should be smart, should be hard worker, and having a strong national and the international network connections for their all over development. So this is a small attempt through this microGB talks, and for that we are very grateful to the Honorable Dr. A.M. Deshmukh sir, President of MBSI, who provide and encourage for this activity. Through the MBSI platform, we are encouraging the students by taking a number of activities related to the microbiology subjects. We are very thankful to the Dr. Daker sir, principal of SSCS. Special thanks to our management to permit us for this activity. Today we have the international speaker, Dr. Retno Kauri ma'am. She is a professor in microbiology department. It's a very golden opportunity for our students to attend her lecture. And I'm very thankful, ma'am, for accepting these invitations and be ready for these lectures. Thank you, ma'am.
Thank you, ma'am. So we are also very much glad to have Dr. A.M. Deshmukh sir. His vision and mission towards the country are the strong reasons sir have always motivated by being an example, by giving an opportunity to organize such events, by sharing his expertise and knowledge. So on this great event, I request Dr. A.M. Deshmukh sir to please give a keynote address and enlighten us with your valuable words. Thank you. Over to you, sir.
Thank you. Thank you very much. I'm very happy to talk in this session. This is the session where all speakers from out of India will deliver a talk. That is International Speakers Talk Series. It has been started by Dr. Daikar and Dr. Sarita Bhutada. Both of them are very, very active. And both of them are responsible for arranging such type of talk. I will also, I'm very happy that this is the first lecture in the series. And this first lecture is being delivered by Retno ma'am from Bali, Indonesia. So as a president of Microbiologist Society, it is my duty to welcome you all in this new lecture series, which will be every Sunday. And it is being recorded. It will be again available for other interested students. Thank you. Thank you very much.
Thank you very much, sir. So now, without further ado, we will turn the time to our today's speaker. So I request Miss Leena ma'am to give a brief introduction of today's speaker. Thank you. Over to you, Leena ma'am.
Thank you, ma'am. Good morning, everyone. I'm Miss Leena Mantala. Welcomes you all again on the behalf of Sanjani Arts, Commerce and Science College for this great international webinar. It is our great honor to have our today's guest, Dr. Dra Retno Kauri. It gives me an immense pleasure to introduce our today's guest. Ma'am hardly needs any introduction. Professor Dr. Retno, she is from the Bali, Indonesia. She is a professor in microbiology. She was the head of microbiology laboratory biology department, FMIPA, Udiana University, 2005 to 2021. She was also the head of Study Center Radioisotope, Udiana University from 2005 to 2010. Ma'am is expertise in subjects such as microbiology and virology, microbiology industry, soil microbiology for undergraduate program, soil and waste microbiology for master degree, and biocontrol microorganism for doctorate degree. Ma'am has given many oral presentation of papers at many seminars or scientific meetings. Firstly, at the 7th International Student Conference at Ibarki University on title Potency of Antifungal Activities of Streptomyces species against the Fusarium oxysporum, the cause of leaf rot disease at Ibarki, Japan on 3rd December 2011. Also, at proceeding enhancing the academic collaboration through a UNINET scientific meeting on title PCR using hypersensitive reaction and pathogenicity specific primer pair of several infected horticulture plants at Udiana University in Bali on 15 Feb 2016. Also on proceeding international conferences of bioscience in Bali on 15th and 16th July 2016. Also, the IOP 6th International Conference on Sustainable Agriculture, Food and Energy, Farm and Environmental Science in 2019 in Manila, Philippines. In 2019. With these great achievements, I request ma'am to share her valuable words by starting over the session. Ma'am, please. I request ma'am to start over this session.
Okay. Good afternoon in Bali and good afternoon in Bali and good morning in India. Thank you for today. First of all, I would like to say thank you very much for inviting me as a speaker today. And also thank you goes to Sanjivani Group of Institutes, Sanjivani Art, Commerce and Science College, Kopargaon, Maharashtra, India. And also many thanks to Dr. Desmukh as a President Microbiology Society India. And also Sri Sumitada, Sri Amit Dada, and Kolhe as a patron from Sanjivani Group of Institute. Thanks go to coordinator Mrs. Nagarkar, Mrs. Mantala, Mrs. Power, and Ms. Gafhail. And also many thanks to organizing secretary, Dr. Sarita and Dr. Dahikar as a convener. Today, I would like to share my research that I have been done. The title of my research is the use of Streptomyces, thermocarboxydus, to control the cause of leaf rot disease on aloe vera plant or Aloe barbadensis Miller in Bali. An aloe plant is 3 different aloe plant, Aloe barbadensis, Aloe ferox, and Aloe peri. In this research, I use Aloe barbadensis Miller. Aloe barbadensis Miller has been planted in Bali since 2006 with the total is 139 farmers in that spread. Hello, have you here?
Yes, ma'am, you are audible, ma'am.
Okay. Aloe vera factory has been built in Bali. The owner is from Holland, and then they collect the aloe plant from 139 farmers and spread out in 5 regency in Bali. As we know, the leaves of the gel of the Aloe barbadensis Miller can be used as a cosmetic, as pharmaceutical, food and beverage. And here we call it Aloe barbadensis Miller. In Indonesian name is lidah buaya. And then with the flower, it's very beautiful flower of Aloe barbadensis and yellow in color. And then have a big leaves. And then, but however, there is a pathogen there that's we call it this leaf rot disease. We can see that the leaves become rotten, dried, and brown in color. This slide shows that 5 regencies in Bali, the plantation of aloe vera is infected by this pathogen. This quite a lot decrease the production of the plantation is about 20%. Actually, the farmer use synthetic fungicides to control or to combat the pathogen. But as we know, the synthetic fungicide has a bad effect for the environment, such as decreased soil pH, pathogen resistant, decreased population, and non-target organism. So we have to find a solution to use biocontrol. One of microorganism to use is I choose a Streptomyces because the Streptomyces can produce extracellular hydrolytic enzymes or antifungal agent, both antifungal or antibacterial. The mechanism to the Streptomyces to combat the fungal is by degradation of fungal membrane. This research is objective is to determine species of Fusarium that found that this leaf rot disease caused by Fusarium. I name it Fusarium sp isolat FO 2010 because I found it in 2010, causing leaf rot disease on Aloe barbadensis milan in Bali. It's about 20% affected by this pathogen. And then the effectivity of Streptomyces isolated from the soil around aloe vera with no disease symptom in inhibiting the growth of the Fusarium. And then to determine the species of Streptomyces that capable to inhibit the growth of Fusarium. And then to analyze the mechanism of Streptomyces in inhibiting the growth of the Fusarium and the structural response of Fusarium on the treatment of Streptomyces culture filtrate. And then the last, my experiment is to study the effectivity of Streptomyces culture filtrate to inhibiting the disease in aloe vera plants in glasshouse experiment. This is the sample collection. This of 5 regions in Bali, Badung, Bangli, Gianyar, Karangasem, and Buleleng. Each region collect 3 plantation, different area, 3 plantation, except in Buleleng, because in Buleleng there is a huge aloe barbadensis plantation. It's about 4 till 5 hectares there. And then the ecological and temperature is very good to grow this plant, but still there is a pathogen there. And then this is the flow of my research. I survey first. I survey for disease on aloe plantation, and then I found the disease, and then I've isolation of the Streptomyces from the soil around aloe barbadensis without symptoms. Why? Because in this plant, aloe barbadensis plant, why the plant can not infected by the pathogen. I think that that time there is a microbe there to protect the plant from the pathogen. So I collect the soil surrounding the plant, healthy plant. And then I did dual culture to know which Streptomyces is the highest inhibitory zone they can produce. Then I did identification molecular, both for fungi and for bacteria. And then I isolate the filtrate from Streptomyces species that has high inhibitor zone, and then continue with filtrate activity test with well diffusion method, and I measure the minimum inhibitory concentration of the filtrate from Streptomyces. And then I have to look mechanism of Streptomyces in inhibiting the growth of the Fusarium, and the structural responses by using scanning electron microscope and transmission electron microscope. I did in Ibaraki University, Japan, for three months. And then the last my research is in vivo test in a glass house. And then next, I have to show you about the this is Streptomyces isolation that I told before. I collected from the soil, the near the healthy plant from the infected plant, and then dilution series, and I grow it in humic vitamin acid agar for 2 weeks at 28 degrees Celsius, incubated. And then cospotulate I done that to confirm, untuk confirmation about the whose that is Fusarium is cause leaf rot disease. Then infected leaves in that picture shows that the postulate confirm that leaf rot disease caused by Fusarium sp.
Ibu internasional jadi gini bisa gede. Ja apalang nih tesnya.
Because the symptoms same with the postulate cough and leaf rot disease in the field. This pathogen infected very fast in the plant of aloe, so this make the farmers decrease the income. Filtered isolation on Streptomyces species. I grew the Streptomyces for 5 days and take the colony, and then take into the I put on into the bottle, big bottle, and then incubation for 14 days at 28 degrees. And then after 14 days, filtration by paper dish filtered size 0.45 and then partition with the N-butanol, and then evaporation, and after that we got it filtered Streptomyces species. Scanning electron microscope preparation at Ibaraki University, Japan. First, I have to the dual culture to make 1% filtered concentration treatment to Fusarium sp. You can see from the photo that there is in the left hand is a control without giving a treatment, and then the right hand is giving a treatment. This is a quite long experiment in there. But I can do it by myself under supervised Professor Nita Yoji and Dr. Takashiyama. And for transmission electron microscope is quite long preparation because I have to cut the membrane cell, the cell into the size 0.45 micromillimeter. That's a little bit very hard to prepare transmission electron microscope. And then we have a result that the percentage of leaf rot disease on aloe vera plant in 5 regencies in Bali. This location, Badung, Bangli, Buleleng, Gianyar, Karangasem, and percentage of disease. In Badung, there is at that moment there is a heavy rain at that time. So in Badung region, there is the disease percentage is quite high, is about 21.60. And then the interesting ones is in Buleleng, as I said before, that in there is very the temperature is very high, so the plant is look very healthy, but still pathogen there is about 2.20%. That average of 3 observation point on each regency and has a standard deviation. This picture shows you about the leaf rot disease caused by Fusarium species. This is a picture is microscopic that contains macroconidia of the Fusarium sp. This morphological is a curve in morphology. And then the B picture is the colony of Fusarium in white colony in color. And this scanning electron microscope, Fusarium sp. isolate FO 2010. This A is macroconidia, B is microconidia, and C is chlamydospora. It's interesting picture for me because from the small we can see by using scanning electron microscope we can see more clearer.
Kurangi terus makan buah, jadi suka apel.
Next, identification Fusarium with 18SR DNA. This shows about the agarose gel electrophoresis, 16SR DNA, Fusarium sp. isolate. Len 1 marker gain ladder 10.
Dia ngasih kuliah
0.1 to 2 kbp, Nipo gain wako. Fragment band 18SR DNA, Fusarium sp. 550 BP. I did in Ibaraki University at Laboratorium of Molecular. And then if we see from the next slide is about phylogenetic sequence of Fusarium library. The size of the bar shows a sequence different of 1%. In here, the isolate Fusarium FO 2010 near close relationship with Fusarium oxysporum. Here is Streptomyees isolate that I can isolate is I got 24 isolate there with different characteristic. With using human vitamin acid agar. Let's continue my correction of the Streptomyees. Then we now talk about the dual culture between Fusarium oxysporum and Streptomyees that has a high inhibitory zone. From the template B, we can see that in the middle is Streptomyees, but and the other side is Fusarium. And we can see that the Streptomyees can inhibit the growth of Fusarium. And the highest can inhibit the Fusarium is Streptomyees GRRK. We can see from the graph that the percentage of inhibitory zone from Streptomyees sp. with Indonesian language with Fusarium oxysporum FO 2010 with 24 isolate with has different inhibitory zone, and the highest is Streptomyees GRRK. Continuous is identification Streptomyees isolate GRRK with 16R rDNA. Produce PCR Streptomyees isolate GRRK using primate 20F and 1000F500R. Lane 1 kb DNA leader fermentase. Lane 2 and 3 band target is 1480 BP. A phylogenetic tree describing the position of GRRK isolate based on the 16S rDNA gain sequence compared to several strains of Streptomyees and Macromonospora. And we can see from the phylogenetic tree that GRRK isolate Streptomyees close relationship with Streptomyees thermocarboxydus stain height HD with total 96%. Continues with morphology Streptomyces thermocarboxydus by using scanning electron microscope. A picture shows that Streptomyces thermocarboxydus taken via scanning electron microscope. A is aerial hyphae and B is conidia with conidia. And then cross section of conidia, I use deep freeze cut method at Ibaraki University, so I can cut the spora. We can see the inside the spora, but we don't know the component of this inside the spore. And then photo of Streptomyces thermocarboxydus conidia taken by Sam. The conidia are spherical in shape with a leafy surface and it's very interesting because we know that the conidia is like a fluffy. And then this is ultrastructure photo of Streptomyces thermocarboxydus conidia taken via transmission electron microscope. The conidia has a cortex, core material, cytoplasmic membrane, and cell wall. And still continues with ultrastructure photo of Streptomyces thermocarboxydus hyphae taken by TAM. This is a ribosome, mesosome. We can see in that ultrastructure photo by transmission electron microscope. Continues with inhibitory test filtrate Streptomyces thermocarboxydus. We saw in the 100% of filtrate can inhibit with the inhibit zone. We compare with the control. And then I measure the minimum inhibitory concentration or MIC. We can see from the table that antifungal activity of several concentration of Streptomyces thermocarboxydus filtrate again Fusarium oxysporum on potato dextrose agar medium. We can see here that the MIC of filtrate Streptomyces thermocarboxydus is 60%. And then microconidia photo of Fusarium oxysporum taken by SEM. This is a control, but and then B is treatment with the filtrate Streptomyces thermocarboxydus. We can see from the picture, the B, that the macroconidia become flat and look like dried. Photograph of Fusarium oxysporum macroconidia after being treated with the filtrate taken through the transmission electron microscope. Perforation of the macroconidia cell membrane, and then this is arrow O is organelles leaving the cells, and then we can see that the macroconidia is broken. And then if we look the hypha, what happened with hypha? Yeah, this is the hypha photograph of hypha of Fusarium oxysporum. In the A is control, but after we give I give a treatment, the the hypha become groove hyphae surface. And then how about if we look by transmission electron microscope, about the hyphae. This is the hyphae normal control with the treatment. So about the hypha of Fusarium is consists of mitochondria, cell nucleus, cell wall, plasma membrane, and ribosome. After I give a treatment with filtrate, ultrastructure photo of Fusarium oxysporum hyphae after being treated with Streptomyces taken through TEM. The A, the cell wall is damaged, and then the B, the plasma membrane of the cell is perforated. You can see clear here in magnification 150,000 times. This is a picture of photograph of Fusarium oxysporum microconidia taken by Sam. Microconidia is an uneven surface with the filter treatment microconidia shriveled and perforated. And ultrastructure photo of Fusarium oxysporum microconidia taken by TEM. We can see in the A is a control without treatment, but after give a treatment, there is the damage to the cell wall and the presence of perforation in the plasma membrane. How about the chlamydospore? This is shows you about the chlamydospore of Fusarium oxysporum. Yeah. This A is a control, but after filter treatment, the chlamydospore with irregular shape and broken. The final test in the final experiment is the test the effectiveness of Streptomyces thermocarboxydus culture against Fusarium oxysporum on aloe barbadensis plant in glasshouse experiment. Here the table shows about the percentage of disease, and then I use to control only give a pathogen, and then I use a chemical fungicide mancozeb, and then STR1 mean I give a spray 0.5 milliliter 1 times, and then I spray STR2 2 times in different days, and then 3-4 give 3 spray and 4 spray in different times in different day. I mean, it has that the STR4 can is not different with mancozeb to combat to inhibit the pathogen. Here is the picture of aloe barbadensis leaves with leaf rot symptom in the glasshouse treatment. I took a picture on that. It is a control. The leaves become rotten and dried in color, and then treatment is STR1, STR2, STR3 still. The pathogen is STR1 still there, STR2 and also, but after give us STR3, the pathogen is can cure by the filter of the Streptomyces. If we compare with MNC, MNC is mancozeb, this chemical fungicide. Result as a conclusion in my research is first, leaf rot disease on aloe barbadensis miller was found in 5 along aloe producing district, namely Buleleng, Karangasem, Bangli, Gianyar, and Badung. The cause of aloe is leaf rot disease, not leaf blast, is the fungus Fusarium oxysporum. And then several Streptomyces isolate that were able to inhibit the growth of the fungus Fusarium oxysporum could be isolated from the soil around the aloe vera without any symptom of the disease. Isola GYRK isolate was the isolate that had the highest inhibition against the growth of the fungus Fusarium oxysporum. Streptomyces GRRK identified as Streptomyces thermocarboxydus, and then the mechanism of action of Streptomyces thermocarboxydus in inhibiting the growth of fungus Fusarium oxysporum is through an antibiosis process, namely through the production of antifungal antibiotic extracellularly with an MIC of 60%. And then the filtrate of Streptomyes thermocarboxydus inhibited the growth of Fusarium oxysporum by damaging the cell wall and plasma membrane of Macroconidia, Microconidia, Hyphae, and Chlamydospore. The treatment of thermocarboxydus culture was effective to inhibit the growth of the leaf rot disease in the glasshouse experiment. Inhibiting capability of Streptomyes thermocarboxydus filtrate was comparable to that produced by synthetic fungicide of Mancozeb. This is the 4 region. The 5 region, I lost my picture. This is the farmer is there, and the bottom and the left hand side, right hand side is the in Buleleng. This is 4 hectares of aloe barbanensis plantation there. So it's very good plantation in Buleleng. Thank you for the farmer there. And then this is the, I have to say many thanks for Professor Nita Yuji. Professor Nita Yuji, if I look in the left picture in the left, is in the, with the tie. And then beside him is Professor Naomi Asagi. And then the picture in the right-hand side is Dr. Takashi Homa, who supervised me for electron microscope. And then Dr. Namikawa is to help me for when I in daily life in Ibaraki University. And also many thanks to all crop production member at Ibaraki University, AMI Japan. This is my last slide. Thank you very much for the opportunity, Dr. Desmo, and the others for the opportunity to me give as a speaker today to share my my research. Thank you.
Hello.
Hello. Yeah.
Thank you so much, ma'am, for your great session on the potency of Streptomyces thermocarboxydus to combat the Fusarium oxysporum, the cause of leaf rot disease on aloe barbadensis male in Bali. Really, ma'am, it was a very great session. The ma'am have highlighted many great points in her experiment. She have first told about the isolations of the Streptomyces, the symptoms shown by that Fusarium species, also the leaf rot disease by Fusarium species, identification of Fusarium, and also told about the inhibitory zone of inhibition in the percentage form, identify of Streptomyces isolates, the morphological characteristics, and the very great results she have achieved. That Streptomyces species isolate, which is identified as Streptomyces thermocarboxydus, the mechanism of action. So, ma'am, it was a really very great session.
Thank you.
For this first lecture of webinar series, we are very glad that we have more than 200 participants who are attending this webinar. So thanks to all the participants.
Yes, ma'am.
Yes.
The session was excellent.
Thank you very much, Dr. Desmo.
Yes, and it was the first lecture in our series.
Yes.
Because this series will continue every Sunday throughout the year. And you have delivered a first lecture in the series, so I congratulate you. May I ask one question, one suggestion?
Yes. Okay.
Your work is excellent.
Thank you.
one of my student also worked on the same line. What I will suggest you, you have studied the Streptomyces species against pathogenic organism.
Yes.
I will suggest you test its phosphate solubilizing activity, nitrogen fixing activity, iron solubilizing activity, and find out whether the same organism can be exploited as a biofertilizer and biopesticide.
Yes.
That's the need now. One organism playing a dual role, you have proved that it is a biopesticide. It can be used.
Yes.
Now you go for biofertilizer activity of the same organism.
Yes.
Okay?
Okay, I will do it.
Yes, yes.
Thank you, Dr. Desmond.
Yes. Any other question, please? I think Dr. Rahman is from UK has joined. Dr. Rahman, please. Please make a comment.
Deshmukh.
Yes.
Yeah, thank you. Can you hear me?
Yes, yes,
Yeah.
yes.
Yes, I hear
Oh,
you.
thank you. Yeah, it's very interesting presentation. And we have been working with Fusarium venenatum, or it's one of the local company. They manufacture a product, food grade product, which is available in most of the supermarket in the UK. So they use very large fermenter to produce this fungal hypha, and then they rupture the cell and then take the protein out of the only the cell wall. They use that to manufacture the product. So it is coming from different direction. Instead of, so it could be the Fusarium, even though it is a pathogen, when the cell is ruptured and then denatured, then that cell wall could be used as a protein, food grade protein in the UK. So maybe in future I'll communicate. I have a full presentation. If you want, I can share that information in future event. Thank you. And
Thank
this, and also it's for the antimicrobial activity, and also for plant, the agricultural application. We are working on the combination of these antimicrobial compound with the nanomaterial to make cosmetics or to replace synthetic agrochemicals. So clearly the work what you have presented, it fits very well with what we have been doing. So I'll be in touch with Dr. Deshmukh and yourself to see what we can do. Thank you.
Thank you. Thank you,
Yes,
Dr.
welcome,
Rahman.
sir. Welcome.
Thank you.
Thank you.
Sheikh want to ask one question. Sheikh. Sheikh has written question in chat box. Lina, read the question.
Yes, sir. Yes, sir. Ma'am, she have asked one question that if she would like to know that if volatile and non-volatile metabolites screened.
The metabolite, I continued this research and have done few months ago that this metabolite. Hang on. I have my new research. I will present it at a microbiology conference. This metabolite of Streptomyces thermocarboxydus I already analysis by using gas chromatography mass spectra. I found that the metabolite active of Streptomyces hexaneoxide and pentane, hexane, butane, and methylene chloride. That all of this metabolite is anti-fungal for the fungi. and hexanoic acid methyl ester as indicator for metabolite active in actinomycetes. This is my new research that I will give a talk at microbiology conference in Bali next 28th of October.
Okay, ma'am. So participants, those who have questions, they may drop a message to chat box, or they may ask to ma'am.
Okay.
Hello.
Yes.
Hello, ma'am. Myself, Kallani. I want to ask the question.
Yes.
Okay. Ma'am, my topic is I'm doing the PhD, and my topic is related to this Fusarium oxysporum. I'm searching the activity that the medicinal plants that are present in our country, that medicinal plants antifungal activity against this Fusarium oxysporum, which is isolated from the soybean plant. So, ma'am, I have taken the isolate from the soybean plant, but I am not able to confirm that those isolates I have obtained that are may Fusarium oxysporum or may not. So, how can I confirm that?
Okay, thank you for your question. You mean you have difficulties to obtain the
Identification.
identification?
No.
Or
Identification. I have identified on the normal light microscope about the morphology structure of that Fusarium. But again, how to means if I want to main to that that is the Fusarium oxysporum. I want to confirm that. So how can I? Can you?
Yeah, I see. You can identify with the molecular approach using
Okay.
gene as our DNA. That
Okay.
you can identify your fungal by using molecular identification. Because if we use only of identification with identification book or with the microscope is not enough, because the color of the hyphae in medium is not enough to get identification from that. So you have to identification using a molecular approach. Thank you.
Okay, ma'am. Thank you, ma'am.
Okay, ma'am. So if there are again any questions, participants can please ask. If there is no questions, so shall we conclude? Sir?
Yes,
Dr.
Lena,
Sarita,
you
ma'am?
can continue.
Yes,
Lena,
yes,
you
sir,
can.
yes, sir, yes. Okay, yes, sir, yes. So I would like to conclude the session by giving a short vote of thanks. So with this, I request Ms. Rutika, ma'am, to please give a vote of thanks. Ma'am, please. Good afternoon. I deem it a great honor to propose the vote of thanks on behalf of Department of Microbiology, Sanjeevani Arts Commerce Science College, Kopargaon, to all who have helped us in making this webinar such a resounding success. First of all, I would like to propose hearty vote of thanks to our distinguished speaker, Professor Dr. Retno Kauri, for making an excellent presentation and making this webinar interesting and meaningful. I thank Dr. Pattenatu Rahman from UK for his valuable presence. I take this golden opportunity to thank respected
Dr. A.M. Deshmukh Sir, President MBSI, for providing us such a great platform for this interactive session. I would like to express our profound gratitude to our principal, Dr. S.B. Dhaikar Sir, for his support in this webinar. I would also like to thank our beloved HOD, Dr. S.A. Bhutada ma'am, for her guidance. I am happy to express vote of thanks to our staff members who have made this webinar a success. I also thank all the respected attendees joined through online mode for their presence. Finally, the wonderful students who have turned up in such great numbers. Thank you so much for your cooperation. Once again, I thank all for your cordial cooperation. Thank you.
Thank you.